Hello everyone. Now in this part we will discuss the complete cell chapter of biology of 9th class in one shot. So let's get started. First we will discuss the discovery of cell. We all know that Robert Hooke in the year 1665 discovered the cell for the first time. What he did? He actually took a slice of cork under his self-made microscope and observed a honeycomb-like structure which he named as cell. This slice of cork is a part of bark of tree, that is the dead part of the plant, that is the dead part of the tree. So the cell discovered by Robert Hooke was dead cell. Now moving on, let us understand the meaning of the name of the chapter, why the cell is called the fundamental unit of life. Here the fundamental means basic or elemental thing by which any organism is made of. So cell is the basic or elemental thing or material by which all living organisms are made up of. Now moving on, we have the definition of the cell, that is cell is the structural and functional unit of life. Why it is called structural unit? Because the body organization of any living organism is made of cells, so it is called structural unit, and it is called functional unit as well, as these cells has to perform all the important functions like respiration, digestion, excretion, etc. So cells are also known as functional unit of life. Now we have the shape and size of cells. The body of human beings or other animals and plants are made of million cells. So the shape and size of cells vary from different organ systems. Suppose in our nervous system, the size and shape of cells are different from the cells found in this muscle cells. So in detail we will discuss the shape and size of cell in the next chapter, that is the tissue chapter. Now we have few facts about cells, that means largest cell, which is the largest cell, ostrich egg, which is the smallest cell, mycoplasma cell, which is the longest cell, nerve cells that are found in our nervous system are called nerve cells. Whenever we study a cell under microscope, we found different type of structures are embedded inside the cell, and these structures are called cell organelles. So let us discuss few discoveries related with the cell and cell organelles. In 1665, Robert Hooke discovered the first cell, that was the dead cells. In 1674, Emil Leeuwenhoek discovered the first living cell from the pond water. In 1831, Robert Brown discovered nucleus. In 1839, Purkinje discovered protoplasm. In 1857, Kolliker discovered mitochondria. However, in 1898, it was Benda who given the name mitochondria. In 1885, Andrew Shimpo discovered chloroplast. In 1953, George Palade discovered ribosomes. In 1945, Porter and Thomson discovered endoplasmic reticulum. In 1898, Camillo Golgi discovered Golgi bodies. In 1955, C. Dedius discovered lysosomes. So, from nucleus to lysosome, all these are cell organelles that are present inside the cell. Now moving on, we have cell theory. Schleiden in the year 1838 and Swan in the year 1839 Schleiden in 1838 and Swan in 1839 formulated cell theory. According to them, cells are the basic unit of life and all living organisms are made of cells. Later on, another biologist, Rudolf Virchow, in the year Rudolf Virchow in the year 1855 also given his view that all cells arise from pre-existing cells. This statement comes from he given a phrase omnis cellula is cellula, that means all cells arise from pre-existing cells. Now let us see the difference between plant and animal cells. Cells found in the plant are called plant cells, while the cells that are found in the animal are called animal cells. So we will discuss plant and animal cell at the last of the chapter. Now moving on, we have uni and multicellular organisms. On the basis of the presence of number of cells, organisms can be divided into unicellular organism and multicellular organisms. If an organism is made of single cell, it is called unicellular, while multicellular organisms are those that are made of many cells. So example of unicellular organisms are amoeba, paramecium, chlamydomonas, etc., while the example of multicellular organisms are plant, animal, etc. In unicellular organisms, a single cell has to perform all the important life processes like respiration, digestion, excretion, reproduction, etc. While in multicellular organism, there is a division of labor in which few group of cells carry out the function of reproduction, while the few groups of cells carry out the process of respiration. So there is division of labor, and in case of multicellular organisms, as there is team effort in multicellular organisms. So in multicellular organisms are more efficient and have more lifespan, while in comparison with the unicellular organisms that are less efficient and have less lifespan. Now moving on, we have prokaryotic and eukaryotic organisms, and the organisms are divided on the basis of nucleus. It is called on the basis of presence of nucleus. Organism can be divided into prokaryotic organism and eukaryotic organism. Pro means primitive, karyos means nucleus. So whenever nucleus is primitive or undeveloped, it is called prokaryotic organisms, and U means advanced and karyos means nucleus. When nucleus is advanced type or developed type, then it is called eukaryotic organisms. Generally, the size of prokaryotic organisms are small, 1 to 10 micrometer, while in eukaryotic organisms are larger as compared to the prokaryotes. Their size may vary from 5 to 100 micrometer. If we look at the nuclear region, their nuclear region are undefined and their nucleus is called nucleoid. Single chromosomes present. Membrane bound cell organelles are absent in case of prokaryotic. Example is bacteria, while in eukaryotes the size may vary from 5 to 100 micrometer. Their nuclear region is well developed, well defined. Nuclear membrane is present. Membrane, more than one chromosome is present. Membrane bound cell organelle, that is mitochondria, plastids, endoplasmic reticulum are present. Human being and other plant and animals are an example of eukaryotic organisms. Now moving on, we have the structure of, we will discuss the structure of cells. Whenever we study a cell under microscope, we would come across three features in almost every cell, and they are plasma membrane, cytoplasm and nucleus. So in this structure, the outermost layer is plasma membrane, the central part is the nucleus, and the part or area between the plasma membrane and nucleus is the fluid part, and this is called cytoplasm. So here first we will talk about the plasma membrane, that is the outermost layer of the animal cell. It is found in both type of plant and animal cell. It separates the cell from outer environment as it is the outermost layer. It is called selectively permeable membrane as it allows or permits the entry or exit of materials in and out of the cell. This plasma membrane is made of phospholipids and proteins, so it is flexible. Now let us talk about the function of the plasma membrane. It performs basically two functions. First function is as it works as mechanical barrier. That means it protects from outer unfavorable conditions. The second main function of plasma membrane is that it helps in the movement of materials across the cell. So let us discuss the movement of how the material enter or exit inside the plasma membrane. So this process is basically of two types: active transport and passive transport. In active transport, movement of materials in the form of gases and liquid takes place from the lower region to the lower region of concentration to the higher region of concentration. So this process requires energy. While passive transport, the movement of materials in the form of liquid and gases takes place from the higher region of concentration to the lower region of concentration, and here no energy is required. This active transport can be of two types, endocytosis and exocytosis. Endocytosis, it is the process of engulfing of food materials by the organisms. This can be of three types: phagocytosis, venocytosis and receptor mediated endocytosis. In phagocytosis, food are engulfed by the organisms in the form of solid materials, while in pinocytosis liquid materials are taken by the cells, and in receptor mediated there is a source that helps in engulfing of food materials. And exocytosis, that is the removal of waste materials by the organisms. The example of this endocytosis is generally unicellular organisms like amoeba. Now moving on, we have passive transport. This passive transport is of two types: osmosis and diffusion. In diffusion, remember that in diffusion there is exchange of gases, while in osmosis there is exchange of water. So in diffusion, movement of gaseous molecules takes place from the region of higher concentration to the region of lower concentration, and the example of diffusion is exchange of respiratory gases in human beings. Concentration of oxygen is higher in outer surrounding, so we inhale oxygen inside our body, and due to the metabolic activities, carbon dioxide that in the form of waste materials are accumulated in our body, and concentration of carbon dioxide in our body at that time becomes higher, so it moves outside where the concentration of carbon dioxide is low. So this is the example of diffusion process. Now moving on, we have the osmosis process. It is a movement of water molecules from the higher region of concentration to the lower region of concentration through a semi-permeable membrane. It's called osmosis process. Generally the plants or you can say root absorbs water and minerals from the soil by the process of osmosis. So let us understand the second organelles that may be present in all type of cells, and this is cytoplasm. Cytoplasm, that is the fluid part present inside the cells. Whenever we observe the cell and under microscope, we found a fluid part inside the cell, and that fluid part is the cytoplasm. It is the mixture of cytosol plus cell organelles. Cytosol is made of 90% water, 10% protein, lipid, vitamin, minerals and carbohydrates, and these cell organelles are located or situated in this cytosols. So next we have protoplasm. It is the combination of cytoplasm plus nucleoplasm. Cytoplasm is the fluid part present inside the cell, while the nucleoplasm is the fluid part present inside the nucleus. So protoplasm is formed by the combination of these two, and it was discovered by Purkinje. Next, moving on, we have nucleus, that is the central part of the cells, present in both type of cells, that are plant cell and animal cell. Only the difference is that in plant cell it is slightly situated or located towards periphery because of the presence of large and prominent vacuoles that are present in the plant cells. In animal cells, generally this nucleus is situated at the center of the cell. Suppose this is the cell, this is the outermost plasma membrane, and inside this fluid part cytoplasm is present, and this central part is nucleus. And if we talk about the structure of nucleus, It is double membrane bound structure and its outer covering is known as nuclear membrane. This nuclear membrane has pores. This pores helps in exchange of materials inside and outside the cells. See the structure present inside the nucleus. Fluid part present inside this nucleus is nucleoplasm, and inside this nucleoplasm another spherical small structure is present, that is known as nucleolus. Ribosomes are synthesized in the nucleolus, and this thread-like structures is called chromatin fibers. This chromatin fibers becomes condensed and thick at the time of cell division, and thus it is called chromosomes. These chromosomes carry DNA, that is the genetic or hereditary materials that are transferred from parents to their offsprings. This DNA is responsible for the transmission of characteristics from the parents to their offsprings. So this is the structure of the nucleus, and now let us see the function of the nucleus. It is called brain of the cell as it controls all the activity of cell. It performs the process of cell division and also carry out cell reproduction. It contains DNA, that is genetic material that are transmitted from the parents to the offspring and are important for the transmission of characteristics from one generation to other. And this nucleus also contains nucleolus that synthesize ribosomes. As we know that ribosome is the protein factory of the cell, it synthesizes proteins. Next, moving on, we have another structure, cell wall. This cell wall is present in only plant cells and it is absent in animal cell. It is non-living rigid structure. It forms outer covering of plant cells made of cellulose, that is a kind of polysaccharides, and the function of cell wall is it gives shape to the cell, it gives strength to the cell, it protects cells from mechanical damage, that means from outer injury. It saves the cell from outer injuries. It gives rigidity to the cells, that means it gives hardness to the cell and also provides turgidity to the cells, that means it exerts pressure on the cell wall. It counteracts endosmosis, that is the more entry of the water. If more water enters into the cell would definitely burst out, so it counteracts endosmosis and thus provides and gives turgidity to the cell to protect them from plasmolysis. Cell wall may have deposition of certain substances, and they are cutin that helps in surface coating of the plant, lignin that gives strength to the plants, and suberin that makes the plant impermeable, that means not allow any material to enter into the cell. Now moving on, we have endoplasmic reticulum. It is the network of sheets and tubules. So generally it exists in three forms, in the form of sheets or flattened sacs, or in the form of tubes, or in the form of oblong or rounded bags that are called as vesicles. So one of the end of this endoplasmic reticulum is attached with the nuclear membrane, while the other end of the endoplasmic reticulum is attached with the plasma membrane. And this endoplasmic reticulum are of two types, RER, that is rough endoplasmic reticulum. Whenever ribosomes are present on the surface of endoplasmic reticulum, it is called RER, rough endoplasmic reticulum, and if these ribosomes are absent on the surface of endoplasmic reticulum, then it would be known as SER, that is smooth endoplasmic reticulum. Now the function of rough endoplasmic reticulum is protein synthesis, as it contains ribosomes, that is the protein factory of the cell. However, in smooth endoplasmic reticulum, lipid and fats are synthesized. Now let us see the function of endoplasmic reticulum. First important function of endoplasmic reticulum is that it synthesizes lipids and proteins that are helpful in building the plasma or cell membrane, and this process is known as membrane biogenesis. The second important function of the endoplasmic reticulum is that the rest of the lipid and proteins function as hormones and enzymes. As we know that enzyme and hormone are important for the various chemical activities. And the third important function of endoplasmic reticulum, as it is in the form of networks of tubules and sheets, it provides cytoplasmic framework to the cells for different chemical reactions. The fourth important function of endoplasmic reticulum is that it is in the form of network of tubules and sheets. That means it has to perform a very important function of transportation of lipid and proteins wherever they are required by the cells. So this was the fourth important function, and the fifth important function of the smooth endoplasmic reticulum is that it is helpful in the detoxification of liver. That means toxins present in the liver are flushed out with the help of smooth endoplasmic reticulum. If you look at the structure of endoplasmic reticulum, this is nucleus, and endoplasmic reticulum is scattered all around the nucleus. So here the small bead-like structure appears. It is rough endoplasmic reticulum. Ribosomes are present on the surface. Here the ribosomes are not present, so it would be known as smooth endoplasmic reticulum. Now moving on, we have Golgi bodies. These Golgi bodies also consist of flattened sacs called cisterns. If we look at this diagram, this is the cell, this is the outermost layer plasma membrane, here is nucleus, and this flattened sacs are called Golgi bodies. Whenever these are arranged over one another parallel to each other, this is called cisterns. This structure called cistern. So this cisterns has spherical structure here. This is called Golgi bodies, and this Golgi bodies also consist of spherical structure Golgi bodies, vesicles, and flattened sacs that are called cisterns. So this Golgi bodies is present in between the nucleus and plasma membrane. One part of the Golgi body is facing towards nucleus, and that part is known as cis phase, while the other deeper part of the Golgi body faces towards plasma membrane, and that part is known as trans phase. So this is the Golgi bodies. Now let us see the function of Golgi bodies. It is main function is storage. Somehow this Golgi bodies have connections with the endoplasmic reticulum. The material in the form of lipid and proteins are synthesized in the endoplasmic reticulum, are further transported to the Golgi bodies, and these Golgi bodies store this lipid and proteins, and whenever they are required by the cells, then they modify this lipid and proteins, packed and dispatched to the different target parts of the cell. Another function constitutes simple conversion of simple sugar into complex sugar, and the third important function of Golgi body helps in the formation of lysosomes. Now moving on, we have mitochondria. Mitochondria is the plural, while the mitochondrion is singular. So this mitochondria is called as powerhouse of the cell because it gives power, it gives fuels to the other cells to perform different cellular activity as well as the life processes. So it is called powerhouse of the cell. ATP generating chemical reactions takes place in this mitochondrion. So it is called mitochondria. So it is called powerhouse of the cell, and ATP Energy is released in the form of ATP, adenosine triphosphate, and this is also known as energy currency of the cell, as it gives energy to the cells for various activities and functions. So, if we talk about the structure of the mitochondria, it is the double membrane bound structure. The outermost covering is flexible and porous. That means pores are present on the outer surface for the exchange of materials. Inner surface is deeply folded and it expands and provides area for the cellular reactions. Reactions के लिए, and inside this fluid part is present. Fluid part present inside the mitochondria is called matrix, and this finger-like projections appears is called crista. So, and the part between the outer and inner membrane is called intermembrane space. So, this is the structure of the mitochondria, and the process of glycolysis takes place inside this matrix. The first important function of mitochondria is the production of energy in the form of ATP. Mitochondria is also known as semi-autonomous body, as mitochondria have its own DNA and ribosomes, so it can replicate and make their own structure. So it is called semi-autonomous body. Plastid, that is the pigment bearing part of the plants. Generally plastids are of two types, chromoplast and leucoplast. Chromoplast is the colorful pigment bearing part of the plant, while the leucoplast is the colorless pigment bearing part of the plant. Chromoplast contains colorful pigments like green pigments, chlorophyll, that is responsible for the function of photosynthesis, in which plants prepare their own food with the help of carbon dioxide, water, sunlight and chlorophyll. So the next is leucoplast, that is the colorless pigment, and in leucoplast food are stored in the form of starch, oils and protein granules. And if we look at the structure of chloroplast, that is the green pigment chlorophyll bearing part of the plant, here it is also double membrane bound structure. Outer covering is flexible and porous for the exchange of materials, and inside this the fluid part present inside the chloroplast is called stroma, and inside the stroma this coin are stored over one another, and this coin like structure is called thylakoids. Chlorophyll pigments are present inside this thylakoids. These thylakoids are enclosed in a special structure called granum, and this thylakoids are attached with each other with a membrane called stroma lamellae for the exchange of materials. So this is the structure of chloroplast, and like mitochondria, this chloroplast also have its own DNA and ribosomes. So chloroplast is also known as semi-autonomous body, as it prepares their own structures. ठीक है, अब ये plastid, is it clear? All right, plastids are present only in plant cell, and plastids are absent in animal cells. Now moving on, we have lysosomes. Lysosome, that is the membrane bound cells filled with digestive enzymes. So it is also called a kind of waste disposal system. This digestive enzymes, lysosome, that is membrane bound cells filled with digestive enzymes, and these digestive enzymes are made of rough endoplasmic reticulum. Point to be noted here is this digestive enzymes is made of rough endoplasmic reticulum. This lysosome is a kind of waste disposal system. It helps to clean the cells by digesting foreign materials, by digesting metabolic waste produced by the chemical reactions and are capable of breaking organic molecules or materials into simpler. And at last, these lysosomes, whenever found any damage to its own cells, it bursts out and the digestive enzymes digest its own cells. So it is also known as suicidal bag of the cell. Now we have ribosomes. Ribosomes are smallest spherical organelles. It is membraneless. That means it is not covered by any layer. It is site for protein synthesis, as we all know, and these ribosomes are found in rough endoplasmic surface of rough endoplasmic reticulum, cytoplasm, mitochondria. These are present in mitochondria as well as in chloroplast. Now we have centriole and centrosome. This centriole and centrosome is again found in animal cell and not present in plant cell. It is membraneless structure, that means it is not covered by any layer or cover and found in only in animal cell. They lie near the nucleus. Structure, if we talk about the structure, appear as two microcylinders. Here is a cylinder and it appears as two microcylinders that are attached with the spindle fibers. At the time of cell division, this microcylinder separates and moves to the opposite to the nucleus. It moves to the opposite pole during cell division. Cilia and flagella, that are basal bodies, are developed from these centrioles that helps in the movement of cells. There is also an important cell organelles, that is vacuoles, that are mostly found in the plant cells and are absent in the animal cells, and if found in animal cells it would be of small size. So this vacuole is the storage sacs for solid and liquid contents. The liquid content is present inside the vacuole is called cell sap, and it consists of different type of sugars, vitamin, minerals, protein, etc. This vacuole is surrounded by another layer that is called tonoplast. Almost 80 to 90% of the cell volume is occupied by the vacuoles. Discussing the different cell structures present inside the cell that are known as cell organelles. Now it is time to discuss cell division in very short as per ninth class. Cell division, it is the process by which new cells are formed in an organisms. So it is one of the characteristics of living organisms that new cells are produced in living organisms. So this new cells, let us see the importance of cell division. First, its importance is it is helpful for the growth and development of an organism. It is helpful in replacing dead and injured or damaged cells in the body. Next, its importance is it forms gametes during reproduction. As a result, young ones are produced. So these are the few examples or importance of cell division. Now let us see cell division can be of two types, mitosis and meiosis. Mitosis generally occurs in body cells or somatic cells. So this division occurs in body cells for growth, repair of damaged cells, replaces dead cells, while meiosis, it occurs in germ cells. Germ cells are found in reproductive organs for giving young ones. Next is it occurs in normal body cells. As we have already seen, it occurs in reproductive cells. Next is one cell division occurs in mitosis, while in meiosis two division occurs. Two daughter cells are formed in mitosis, while four daughter cells are formed in case of meiosis. And these two daughter cells are identical to parent cells, while in meiosis the four daughter cells that are formed are different from each other. So this is all about cell division. Now moving on, we have difference between plant cell and animal cells. So first let us see plant cell. Plant cells have cell wall. In animal cell wall is absent. Plastids are present in plant cells while absent in animal cells. Golgi bodies are found as subunits called dictyosomes in case of plant cells, while in animal cells developed Golgi bodies are present. Next is centrosome is absent in case of plant cell, while in animal cell centrosome is present. Vacuole is present in case of vacuole is present in case of plant cell, while it is absent in animal cell. Now look at this diagram. This is the plant cell. It is usually hexagonal in structure. So the outermost layer is cell wall. Next to the cell wall, this is the plasma membrane, and next to the plasma membrane, cytoplasm is present, that is the fluid part. Inside the fluid part, this is large and prominent vacuole, and this is nucleus that slightly moves towards the periphery because of the large and prominent vacuoles. Nucleus is surrounded by endoplasmic reticulum. This is mitochondrion, this is chloroplast. This dot-like structure is called ribosomes. This is vacuole. So this is about plant cell. Now we have animal cell. So the shape of animal cell is almost cylindrical, and this ciliar flagella like structure is called microvilli. This is the outermost layer, that is plasma membrane. Next to the plasma membrane, the fluid part is present, and this is cytoplasm. Inside the cytoplasm, nucleus is located at the center, and it is surrounded by the rough and smooth endoplasmic reticulum. Next to the endoplasmic reticulum, Golgi body is present between the nucleus and plasma membrane. Here mitochondrion is present. Chloroplast is absent in case of animal cell. A new structure centrosome is present in animal cell. Ribosome dot-like structures, ribosomes are present, lysosomes is present. Lysosomes are found both in plant and animal cells. So this is all about the difference between plant and animal cells. So this is all about cell, the fundamental unit of life, in one shot. I hope you like the video. Thank you.